I'm Maria Mcsharry. I'm a graduate student in the Leroux Lab at the University of California, Berkeley, where I endeavor to understand how synonyms can shape genetic messages. Proteins constitute the structure and machinery of cells, and at the center of biology is a molecular process that's concerned with the making of proteins. This consists of transcribing DNA to RNA and then translating RNA to proteins. You will have heard of these processes referred to as gene expression. RNA is translated into amino acids in 3 nucleotide blocks called codons. And you'll notice that the system is degenerate. So a single amino acid may have up to 6 synonymous codons encoding it. Just like language, swapping synonyms in this context may not impact the message, but in other cases, it might render the message less sensical. For RNA translation, changing a codon can change the way that the ribosome moves across a transcript. So it can slow it or speed it up, and that might impact the folding and function of the protein ultimately. Already in my work, I've demonstrated that when we swap slow for fast codons in a yeast metabolic protein, that this change has enough of an effect on the protein that it impacts yeast at an organismal level and it depresses their growth. So we already know that changing a synonymous codon can impact a protein, but the question remains, what determines which codons are used in what context? In other words, we don't yet know how to predict the phenotype that will result from a synonymous codon change. To get at this question, I'm using a yeast gene called YOR1, and it's a homolog to the human cystic fibrosis gene CFTR. In fact, when it is mutated, it has a similar phenotype to the CFTR gene. So I'm tagging this gene with a fluorescent protein either fused to or uncoupled with it in translation. And by switching every single codon in this gene to its synonyms, I can measure the impact of protein output by fluorescence. I've been testing various aspects of my study system, and on the top left panel, I'm showing you here that mutant YOR1 is distinguishable from wild type. You can see that yeast with mutant YOR1 in red fluoresce less, indicating less protein is produced. And I expect that any impactful synonymous changes that I make to this protein will fluoresce at a level somewhere between the wild type and the mutant. If you look at the bottom left, I have demonstrated that uncoupling the translation of YOR1 from the fluorescent protein points to translation as the step most impacted by mutation. And you see this because here, the mutant is more comparable to the wild type because the fluorescence has been uncoupled. I'm highlighting that observation by plotting the mean fluorescence on the plot on the right. Stop by my poster so we can chat more about how the system works, what I expect to find, and how the scope of my project might impact your future. I'd like to thank ARCS for supporting my work, and I encourage you to check out our work at the following websites. I look forward to seeing you at the symposium in April.